Thank you for the introduction. I would also like to thank Prof. Goh and the organizers for the opportunity to speak at this workshop. I'm Joel, Technical Application Specialist for Azenta. In line with the theme of RNA-seq and bioinformatics, I would like to touch on single-cell RNA-seq. Today, I would like to cover a few aspects of single-cell RNA-seq, which I have summarized into a brief history, the advantages of single-cell RNA-seq, and the workflow. So let's start with the history of single-cell RNA-seq. As the name suggests, single-cell RNA-seq is sequencing RNA from a single cell. While RNA extraction has been performed routinely for populations of cells for sequencing, single-cell RNA-seq was limited by the low amount of RNA per cell. Estimates of the RNA per cell ranges from 10 to 30 picogram. So researchers first developed the method for sequencing one cell in 2009. And as you can see from this figure B, as the technology advanced, the number of individual cells that can be sequenced in one study has steadily increased. Among the enabling technology, improvements in single-cell isolation such as microfluidics or droplet-based methods, improvements in the chemistry of amplifying RNA, improvements in multiplexing methods, integration of different methods into automated systems, and improvements in sequencing technology have played a huge role in advancing single-cell RNA-seq. Greater accessibility to single-cell RNA-seq was made possible by the first commercial platform, Fluidigm C1. And the advent of 10x genomics in 2017 accelerated the adoption of single-cell RNA-seq with their high-throughput platform that can interrogate more than 100,000 cells per chip. Moving on to the next question. What are the advantages of single-cell RNA-seq over the standard RNA-seq of cell populations? Here's a table that neatly summarizes the advantages of single-cell RNA-seq. Both bulk and single-cell RNA-seq can examine the differences of transcriptomics between wild-type and mutants, or treated versus control. Both are high throughput and sensitive, and both can identify novel transcripts. However, single-cell RNA-seq can identify rare cell types in a cell population. It can identify mutation in individual cells. It does so by profiling the transcriptome of individual cells. And in so doing, it can determine the heterogeneity in the cell population we are sampling. It can be used to track antigen-specific cells and identify the receptors on immune cells, such as T and B cells. So what is the underlying biology that justifies the need for single-cell RNA-seq? The key word here is heterogeneity. As we all know, single-cell population in any given system is heterogeneous. This is true even in clonal cell populations. If you have performed flow cytometry staining using antibodies before, you will recognize the histogram of the protein expressed on a cell population. This can be CD19 protein on B cells or CD8 protein on the clonal population of ovalbumin-specific CD8 T cells, commonly known as OT1s. At the population level, the histogram reflects the varying degrees of protein expression at any given time. Some individual cells express low levels of this protein, whereas some express a lot, even though they are the same clonal population. So there are various reasons for heterogeneity in cell populations. Firstly, there are non-genetic sources of heterogeneity. This arises from the underlying stochastic nature of cellular processes. For example, the transcriptional machinery transcribes genes in a stochastic manner, switching from inactive to active state and vice versa, leading to transcriptional bursts of the genes rather than a continuous expression of gene transcripts. So while the process of transcription is regulated and ordered, at the molecular level, there is inherent randomness from the laws of physics that govern these processes. There are also spatial differences in where the cells are located in a cell population or an organ, resulting in varying degrees of accessibility to environmental cues, such as microbial interactions, cytokines, and growth factors. There are also temporal differences, such as cell cycle, where the cells are in different stages of the cell cycle. Heterogeneity also comes from genetic sources, for example, random mutations. And the best known example would be cancer, and how after treatment, a drug-resistant cancer cell can escape and cause a relapse in patients. Because of the transcriptional burst during gene expression, this gives rise to the gene distribution patterns that we commonly see in single-cell RNA-seq data. And here is one example. In this part, figure C, the histogram shows the expression levels of gene X on the x-axis and the number of cells sampled in the y-axis. And some cells may have a low or no expression state of gene X, and some cells may have a high expression state of gene X. And if we were to sample the population as a whole using bulk RNA-seq, we may see a normally distributed gene expression profile with a mean of between mu1 and 2. And that is not a true representation of the expression profile. Related to this transcriptional burst, another feature that is commonly observed in single-cell RNA-seq data is zeros. Lots and lots of zeros. So many that is commonly referred to as zero-inflated data, which means more zeros than expected from a statistical model. And various zero-inflated statistical methods were and are still being used to analyze single-cell RNA-seq data. So this zero inflation is still a debated topic and is subject to ongoing research. But for the purpose of this workshop, I would like to provide a brief overview of what this zero inflation is and why is it important to understand zeros in this data. So here we have 6 genes in a single cell. Gene 1 is not expressed in the individual cell due to its lineage, but the other 5 genes are. Gene 2 is expressed but not transcribed at the time of cell isolation. So gene 1 and 2 are true biological zeros. Gene 3, 4, 5, and 6 are expressed and transcribed by this cell at the time of cell isolation. However, gene 3 use a zero count, and this is due to the limitations of the cDNA synthesis steps, and this is a technical zero. Gene 4 is expressed but not amplified, leading to low abundance. Gene 5 is expressed but transcribed at a low abundance. Gene 5 is reverse transcribed and amplified. However, due to the limitations in sequencing, the amounts of gene 4 and 5 are insufficient to yield any reads, leading to a zero. So both gene 4 and 5 are sampling zeros. And in contrast, gene 6 managed to yield sufficient reads to give non-zero data. As you can appreciate from here, the importance of identifying true biological zeros that confer meaning to our understanding of the cell lineage, which is gene 1, and the importance of understanding the biological state of the cell, which is gene 2, and the importance of removing non-biological zeros that arise from the limitations of our technology. Now, having walked you through the nature of single-cell RNA-seq data, let's dive into how single-cell RNA-seq is achieved. So the most popular and widely adopted platforms for single-cell RNA-seq currently are from 10x Genomics and Illumina. We will just go through the 10x Genomics, as others in this workshop probably has covered how Illumina technology works. Where does 10x Genomics feature in single-cell RNA-seq? It provides the platform and reagents to isolate hundreds of thousands of single cells, perform reverse transcription, amplification, and library preparation for all the isolated single cells. The sequencing is then performed on the Illumina platform. And how does this 10x Genomics single-cell isolation work? Their method is similar to JobSeq, but with a higher capture efficiency. The cells are funneled through microchannels and together with the reagents, encapsulated with oil. One oil droplet contains 0 or 1 cell and reagents. Essentially, the oil droplet is the test tube for performing the next step, reverse transcription. In this oil droplet, the reagents contain barcoded primer gel beads, which 10x calls gel bead-in emulsion technology, or GEM for short. So these GEMs contain sequencing adapters, barcodes that index the GEM, which allows each cell to be barcoded with this GEM index, barcode to index the mRNA molecules, which is called the UMI, or Unique Molecular Identifier, and the oligo DT to capture the poly A tail of mRNA. So why do we need unique molecular identifiers? The UMIs are unique sequences used to tag individual mRNA or DNA fragments before amplification. So let's consider a situation. If we have 5 amplified cDNA with the same sequence, we cannot know if this originates from a single source of transcript or multiple sources of transcript. Because the original mRNA molecules are tagged with UMIs before amplification, we can tell the source of these sequences. If it is from a single source, these are PCR duplicates, and we should condense this sequence into one source of transcript. If these are from 5 different mRNA transcripts, we can assign them to the 5 different sources of transcripts. Hence, UMI enables us to accurately quantify the expression levels of genes in single-cell RNA-seq experiments. Now let's briefly run through what happens in the oil vesicle after cell lysis. The mRNA transcripts are captured by the poly DT. Reverse transcription is then carried out in the oil vesicle. Template switch oligo, or TSO, priming then occurs. The TSO is an oligo that hybridizes to untemplated C nucleotides added by the reverse transcriptase during reverse transcription. The TSO adds a common 5' sequence to full-length cDNA that is used for downstream cDNA amplification. Chilean magnetic beads are used to purify the first-strand cDNA from the post-GEM-RT reaction mixture, which includes leftover biochemical reagents and primers. So after amplification and cleanup, we then construct the library. Enzymatic fragmentation and size selection with SPRI select are used to optimize the cDNA amplicon size. The ends are repaired, and A is added to the tail. Read tool is ligated into the amplicons. Further priming then adds the sample index, P7 and P5 into the amplicons. So the final library will contain P7 and P5 for breach amplification during Illumina sequencing. So after sequencing, Cell Ranger workflow provided by 10x is used to process single-cell RNA-seq output, starting from the raw sequencing data, BCL files, up to visualization. The Cell Ranger demultiplexes the BCL files for each flow cell directory into FASTQ files. To determine whether barcode sequence is correct, Cell Ranger compares the 10x barcodes to the known barcodes for a given assay chemistry, which are stored in a barcode whitelist file. The alignment step, the sequences with the valid barcodes are trimmed. The TSO sequence is trimmed from the 5' end of read 2. Poly A is trimmed from the 3' end prior to alignment. And to align the sequence, Cell Ranger uses an aligner called STAR, which performs sequencing splicing aware alignment of reads to the genome. Then Cell Ranger then uses the transcript annotation GTF to bucket the reads into exonic, intronic, and intergenic, and whether the reads align confidently to the genome. Cell Ranger further aligns confidently mapped exonic and intronic reads to annotated transcripts by examining their compatibility with the transcriptome. Any reads that map in the sense orientation to only a single gene is carried forward to UMI counting. Following alignment, Cell Ranger attempts to correct for sequencing errors in the UMI sequence before counting UMIs. Reads that were confidently mapped to the transcriptome are placed into groups that share the same barcode, UMI, and gene annotation. So each observed barcode, UMI, and gene combination is recorded as a UMI count. And the output you will get is the UMI count matrix. And from here, we can filter the data by identifying cells based on the RNA content using Cell Ranger, or we can export the data to SURET and perform this step. So we start by reading in the UMI count data from Cell Ranger into SURET. The SURET is an R package designed for QC analysis and exploration of single-cell RNA-seq data. The count matrix, which we imported in, serves as a container that contains both data clustering and dimensional reduction analysis. Similar to Cell Ranger, SURET allows you to easily explore QC matrix and filter cells based on any user-defined criteria. And here are a few QC matrix commonly used by the community. And these include the number of unique genes detected in each cell. For example, low-quality cells or empty droplets will often have very few genes, or cell doublets or multiplets may exhibit an apparently high gene count. It can also be the percentage of reads that map to the mitochondrial genome, since low-quality or dying cells often exhibit extensive mitochondrial contamination. After QC, we perform a global scaling normalization method. And what it does is that it normalizes the feature expression measurements for each cell by the total expression, multiplies this by a scale factor, and log transforms the results. And because the single-cell RNA-seq data is large, complex, noisy, and has a high number of dimensions and comparatively lower number of samples, single-cell RNA-seq data analysis typically include a combination of dimensional reduction and clustering methods. Dimensional reduction techniques are used to reduce the data to lower dimensions while preserving as much of the structure or information from the higher dimensions. Now, SURET is able to perform the most commonly used ones, and this include PCA, TSNE, and UMAP, which we will cover in the next slide. Clustering methods aim to group data objects into multiple clusters, so the objects in the same cluster are similar and different from objects in different clusters. SURET performs a k-nearest neighbor graphs that is based on Euclidean distance in the PCA space, with the edges drawn between cells with similar feature expression patterns, and then partitions this graph into groups of cells through an iterative algorithm into clusters. And to find differentially expressed genes, by default, SURET uses the Wilcoxon rank sum test to compare a single cluster to all other cells. The Wilcoxon rank sum test is by far the most commonly used method to identify differentially expressed genes in single-cell RNA-seq data. Other types of comparisons are possible, and these methods include DEXIC2, which uses a negative binomial distribution, MAST, which uses a generalized linear model framework that treats cellular detection rates as a covariate, and SURET provides this test as alternative. Here are some visualization tools that I mentioned earlier that are commonly used to represent single-cell RNA-seq data. The first one on the left is the PCA, Principal Component Analysis. And PCA is a time-tested method for dimensional reduction. It is an unsupervised deterministic algorithm used for feature extraction as well as visualization. It applies a linear dimensionality reduction technique where the focus is on keeping these similar points far apart in the lower dimensional space. And it transforms the original data to a new data by preserving the variance in the data using eigenvalues. TSNE is commonly used to visualize single-cell RNA-seq data. TSNE applies a nonlinear dimensionality reduction technique where the focus is on keeping the very similar data points close together in lower dimensional space. It preserves the local structure of the data using student t-distribution to compute the similarity between points in the lower dimensional space. And UMAP is a new kid on the block. It is very similar to TSNE, and the advantages of UMAP include preservation of both local and most of the global structure in the data. And it requires shorter time to compute. While TSNE moves the graph point to point from high to low dimensional space, UMAP makes a fuzzy but topologically similar graph and compress it into a lower dimension. So both Disney and UMAP are used as visualization tools for single-cell RNA-seq. Neither provides information on clustering or variance in the data. If we were to ask which is better for single-cell RNA-seq, the short answer is both are similar. And you can do both for your to show your results in the different manner. And the last plot I would like to introduce is the Violin plot. A Violin plot is a hybrid kernel density plot and a box plot. It visualizes the distribution of the data while showing the summary statistics such as the mean, median, and the interquartile ranges. And Violin plot is commonly used to represent bimodal distribution seen in single-cell RNA-seq analysis. And finally, I would just like to end off with how a Xenter can enable your single-cell RNA-seq analysis. So first, you can provide the cell samples to us. These cell samples can be fixed with paraformaldehyde or methanol. Fixed samples can be shipped at room temperature. Alternatively, frozen cell samples or cell nuclei are accepted. If you have already isolated the cells and prepared the library using in-house 10x platform, submission of single-cell libraries is also accepted for sequencing. And after we have sequenced your library on the Illumina platform, bioinformatics analysis are performed using the cloud-based platform Rosalind. And the Rosalind single-cell gene expression platform enables visual exploration and self-investigation of experimental results, and it gives you the freedom to annotate clusters, visualize cell type arrangements, adjust cutoffs, add comparisons, apply covariate corrections, and find patterns across multiple data sets. This allows you to focus on discovering important features in your single-cell data sets without the steep learning curve for bioinformatics expertise. And with this, I would like to end off today's presentation on single-cell RNA-seq. I hope I have given you a taste of what single-cell RNA-seq entails, and perhaps encourage you to think about how single-cell RNA-seq can be applied to add greater depth and clarity in your research. And please feel free to contact me if you have any questions or would like to discuss how to apply single-cell RNA-seq to your project. Thank you.